Topic: Sexual reproduction in flowering plants. Flowers are the seat of sexual reproduction in angiosperms. In the flower, endoecium consisting of stamens represent the male reproductive organs, and gynoecium consisting of pistils represents the female reproductive organs. A typical anther is bilobed, dithecous, and tetrasporangiate. Pollen grains develop inside microsporangia. Four wall layers, the epidermis, endothecium, middle layers, and the tapetum surround the microsporangium. Cells of the sporogenous tissues lying in the center of sporangium undergoes meiosis, that is termed as microsporogenesis, to form tetrads of microspore. Individual microspores mature into pollen grains. Pollen grains represent the male gametophytic generation. The pollen grains have a two-walled layer, the outer exine and inner intine. The exine is made up of sporopollenin and has germ pores. Pollen grains may have two cells, a vegetative cell and a generative cell, or three cells, a vegetative cell and two male gametes, at the time of shedding. Pistil has three parts: the stigma, style, and the ovary. Ovules are present in the ovary. The ovules have a stalk called funicle, protective integuments, and an opening called micropyle. The central tissue is the nucellus, in which archosporium differentiates. A cell of the archosporium, the megaspore mother cell, divides mitotically, and one of the megaspores forms the embryo sac, the female gametophyte. The mature embryo sac is 7-celled and 8-nucleate. At the micropylar end, the egg apparatus consisting of two synergids and an egg cell. At the chalazal end are the three antipodals. At the center of large central cell with two polar nuclei. Pollination. Pollination is the mechanism to transfer pollen grains from the anther to the stigma. Pollinating agents are either abiotic like wind and water or biotic like animals. Pollen-pistil interaction involves all events from the landing of pollen grains on the stigma until the pollen tube enters the embryo sac when the pollen is compatible, or pollen inhibition when the pollen is incompatible. Following compatible pollination, pollen grains germinate on the stigma and the resulting pollen tube grow through the style, enter the ovules, and finally discharges two male gametes in one of the synergies. Angiosperms exhibit double fertilization because two fusion events occur in each embryo sac, namely syngamy and triple fusion. The products of these fusions are the diploid zygote and the triploid primary endosperm nucleus in the primary endosperm cell. Zygote develops into the embryo and the primary endosperm cell forms the endosperm tissue. Formation of endosperm always precedes the development of the embryo. The developing embryo passes through different stages, such as the proembryo, globular, and heart-shaped stages before maturation. Mature dicotyledonous embryo has two cotyledons and an embryonal axis with epicotyle and hypocotyle. Embryos of monocotyledons have a single cotyledon. After fertilization, ovary develops into fruit and ovules develop into seeds. A phenomenon called apomixis is found in some angiosperms. Particularly in grasses, it results in the formation of seeds without fertilization. Apomix have several advantages in horticulture and agriculture. Some angiosperms produce more than one embryo in their seeds. This phenomena is called polyembryoni. Thank you.